Hey, Ben.
Hey, Lindsay.
How you doing?
Good. You know, um, so, um, yeah.
We'll wait a couple of minutes for people to join.
Hi guys,
Hey, Carmela.
Oh, hello, Carmela.
how are you?
Doing okay? So are you local to us?
I'm local now, yes, for another week, and that's it.
Good.
I wanted
Hi,
Yeah,
Carmela.
some people go south for the winter, and you
I go
go.
east, looking for warmer climate.
Danielle and I have been doing sculling on the Charles. We've decided to try to take that up, which is kind of fun. So, when you go on the Charles Bridge Road, that beautiful community rowing boathouse,
Yes,
we've taken a novice course there, and neither of us has fallen in yet.
you mean sailing?
Rowing.
Oh, rowing, yeah, okay.
So,
How many people in the canoe?
well, it's called a skull. One or sometimes both of us, depending on which one we take.
Mm-hmm. But
So,
it's like 10 people or just two?
just two. When you go across the Charles River and you see all those people on those boats.
Yeah.
So we're a little upstream, but that's what we're doing now. It's just kind of a fun, different thing to take up in our, as a
Great.
couple.
Good luck.
Thank you. Yeah, exactly. That we don't fall in.
All right.
Can you hear me?
We could spend the rest of the meeting talking about sculling, but let's get started. So I want to welcome everybody to our first Precision Medicine and Alzheimer's Disease ARC meeting for the year. We have an excellent slate of speakers all lined up. And before I introduce Ben, just a couple of things about the ARC. There will be, for those of you who aren't familiar with Evans Research Days, part of the Department of Medicine's annual two-day festival, so to speak, to celebrate research accomplishments. And there are distinguished speakers who are invited. But on one of those days, there is an event called Shining Lights on the Arcs. And and we are fortunate because we are a graduated ARC that we've been invited to make a presentation, and you should all attend. I think it'll be virtual as well as in person. Unfortunately, because we're a graduated ARC, we only get 5 minutes to highlight one aspect of our accomplishments. So stay tuned and we'll give more about that. I think I'm going to stop there for right now so we can move forward. So I'm delighted to introduce our speaker today, who is Ben Wolozin from the Department of Pharmacology, and now it's Pharmacology, Physiology, and Structural Biology. Is that the name of your department now?
No, I switched to anatomy and neurobiology.
Oh, well, so scratch that. So now Ben is a departmental colleague of Rhoda and others in the Department of Anatomy and Neurobiology. Ben is a stalwart here at BU, been toiling in the Alzheimer's disease research field for almost as long as I have, and that's a long time. And Ben has numerous accomplishments over the years, published many high-profile papers in top-tier journals. And today we are going to benefit from his words on one of his projects, and it's on RNA of all things. So Ben, do you want to share your screen?
Yeah, sure. And I'm doing it from my laptop because for some reason the PowerPoint on my desktop wasn't working. So
Okay.
everyone
So
see it?
you're all set.
Great. So for a while now, the lab has been strongly focused on looking at methylated RNA. And so I'm going to tell you our update on this. I admit to not being quite as far as long as I thought I would be, because it turns out getting CSF is really, really slow.
Ben, can I just,
Yeah.
I'm sorry to interject, but your title slide here still has your former department.
Good point. Okay, let me change that real time here. That's how recent it is. I will change my title slide. Let's see, get out of here.
So now I don't feel so bad.
Okay. Anatomy and neurobiology. Neurology. Okay. Now here we go. Okay, let me try again. Good point, Lindsay. Okay, there we go. There. All right, corrected. And as everyone knows, I have a conflict of interest because I'm a co-founder and CSO of Aquina Pharmaceuticals. Lindsay, because you used the word toiling in Alzheimer's in your intro, I thought I would just end with a slide that I showed it in my anatomy and neurobiology talk that we're going to be presenting hopefully at CTAD if they accept the abstract, and I think it's promising. So this kind of, the backdrop here is that this comes out of this this idea of what causes a lot of pathology, which is it arises from this this biophysical biophysical process called liquid-liquid phase separation. You don't really need to understand it. It's not totally relevant here. But it's just the idea that RNA binding proteins and tau both form droplets in cells. That's how they organize themselves. But because they have a tendency to go through conformational changes that converts them from a liquid state to a solid state, you get aggregates. And it's the formation of droplets that puts them in relatively high concentrations near each other. So the droplets are important because they control a lot of RNA metabolism, and they also control translational activity at synapses, and they control the stress response. And in many other degenerative diseases, RNA mutations in RNA binding proteins give rise to are linked to the disease. Less so in Alzheimer's disease, because in order to get Alzheimer's disease, you really need to get amyloid, or it isn't, it's not the disease. And so the two relevant RNA binding proteins are RBFOX1, which is thought to regulate amyloid metabolism, and perhaps KELP2, KELP1, although it's also in another locus linked to AD, so that might not be a real hit. But what you can see is you see RNA binding proteins and co-localizing with tau in human brain. Yellow shows the co-localization in these cases, and it's more so for some RNA binding proteins like TIA1, less so for proteins like G3BP, which turns out doesn't like methylated RNA, which you'll see later. But that says that there's this link between tau and RNA metabolism. And so kind of going over older data, we went and developed a Cry2 optogenetic tau construct that could induce tau aggregates, actually membraneless granules in cells over 8 minutes, which we published a couple of years ago. And we converted that to do proteomics. And when we did the proteomics, when we hit it with light, caused formation of tau oligomers, pulled down the tau oligomers and did proteomics, what you could see is that the top binding protein was HNRMP A2B1, which when you do advanced informatics and happen to look in PubMed, the very first papers and a lot of the papers that pop out in this say that it regulates M6, it regulates M6A RNA metabolism, and that kind of happens, is changed during stress. So that led us down a rabbit hole of looking at M6A, which has turned out to be very exciting and very productive. Admittedly, I was hoping I'd have like a lot of kind of more CSF tests to show you, but we do have data where I'm leading up to, and you'll see totally where this is going. And today, I think we're supposed to be receiving 40 samples from MGH. So when we look at the brain using this antibody to M6A, what you can see is there's a stage-dependent increase in labeling with the antibody. I'll mention this later. There's a caveat that we understand now, which is that the antibody is specific to M6A, but as I'll show you later, the actual sequence recognized by the modifiers are broader than that. And so it's likely that this antibody is not specifically specific to M6A. It also recognizes dimethyl adenosine, M6AM. And so we have plans in the work to analyze by mass spec what the antibody is actually recognizing, so we understand this. And we have second generation tools that we're developing. So when you look at this, in order to understand the system, and it becomes important to understand where we go with this, you have to understand the biology of it. So there's these things called writers that add the modifications to the RNA, and the writers there's a complex, middle 3, 14, W, TAD, and RBM15, which are the main writing complex. And I'll show you briefly later that if you knock down metal 3 or metal 14, you can reduce M6A, and that in itself can modify the disease process. Importantly, when you say what actually recognizes the M6A, there are all these proteins that have a YTH domain. And so we'll be showing you a lot of slides that relate to this, because you can take this YTH domain, turn it into a tool, and use it to identify M6A. And then proteins like ALK-BH5 and FTO get rid of it. One other protein, yeah, that's good enough for now. Jenna Libera and Erica Busso a while ago did a study and showed that if you look in a P301S tau mouse and look at metal 3, it's slightly increased with disease. And if you look at like ALK-BH5, it's slightly decreased, which can give rise to an increase in M6A. We have repeated that now in an APP tau transgenic cross and seen largely similar results. So that gives you an understanding of why M6A might be increased with disease. So, but the key here is total M6A, not so informative. It's like saying phosphorylation is increased in Alzheimer's disease. It tells you something, but what you really want to know is what is phosphorylated. So in the same way, we want to know what genes are methylated. So we're in the deep in the process of doing this with the help of Kate Myers from Duke and Xiaoling Zhang's group doing the informatics for us. And we used two approaches. The first approach, which I presented at the last ARC meeting, is using the M6A antibody to simply pull down methylated RNA and whatever else comes with it, and then sequence that. And I'll show you some data from that. But Jenna Libera in my lab has kind of pulled up a new method called DART-seq. And what that is if you take this, if you look at M6A, these YTH proteins identify not just the M6A, but the C next to it and this kind of 6 base context that they use for binding to the M6A. So that gives you a much more highly specific recognition site than just M6A itself. So Kate Myers at Duke took this idea and took the YTH protein, added an enzyme called ApoBec to it, which converts the C to a T, and then use this to do something that she calls DART-seq, to sequence the M6A. And the way this works is, you know, if you have a cell and you know its reference transcription annotations, and then you incubate the RNA with this YTH ApoBec construct, and you can do this either by transfecting cells or using recombinant YTH ApoBec protein. And so I'm not going to show these slides, but we have now made this in the lab, and so we're kind of ready to roll in terms of doing the sequencing on human brain samples. But from that, you then go and just look and say what what bases have been changed when you see a T next to an A, and that can tell you that is a methylated site. So this is important because it, as mentioned, it increases the specificity over the antibody, and the field itself is largely shifting towards this as a tool. It is much easier to do. Instead of requiring many steps, this is kind of a one-stop reaction. Just get the RNA and add the YTH ApoBec, and so that decreases the workflow. And just makes it much easier to go through. And also in the process, you immediately identify the site where the methylation occurs. So there's a lot of information that comes out of this. And so Jenna looked at this and looked at the, as kind of proof of concept, looked at the ZIS gene, did this with HEK293 data, and kind of looked at a site that was known to be highly methylated to see if she could repeat that data. And this is what it looks like. So this is kind of transfection of the DART-seq construct, wild type, or there's a negative control mutant into the HEK cells, and then sequencing. And what you can see in this site is that these bright green are things that are methylated. So you can see that without the DART, there's no methylation. With the wild type DART, there's strong methylation, and then you can use a mutant DART that's inactive, and you don't get any methylation. So, sorry, the YTH itself has been, it's not the enzyme that's inactive, the YTH has been modified, so it doesn't recognize M6A. So there you get much less methylation. And if you look at genes that are not thought to be methylated, they aren't. Sites that aren't methylated, they aren't methylated. So it's nicely specific. So we've now done this on our APP tau transgenic mouse. And the first thing we've done, and what I'll show you, is we've done both the M6A RIP-seq and the DART-seq. So here's the RIP-seq data that we've done. So you can see that we have a large number of targets that we've gotten. We have a lot of significant hits that we're looking at. And when you kind of go and look and see what you get from this, what you can see, so here's kind of what you do is you look at kind of whether the gene expression is changed. And I'll show you specific examples of this, or the methylation is changed. And so what you're looking for is if the methylation in a classic case, the methylation, if it goes up, gene expression goes down because it stimulates degradation. The methylation stimulates degradation of the RNA. However, what we find is that in a lot of cases, the methylation goes up. There's no change in the gene expression. And we believe that that is because we're changing the localization of the RNA. And so we're doing experiments now to test that, looking at synaptosomes and also using RNA scope. But what you can see is that when you look at all these genes and what's changed, what you can see is that the changes in methylation overwhelmingly occur in genes that regulate synaptic processes. So that suggests that that's consistent with what we saw with the M6A antibody, where the changes were in neurons, and that suggests that these are regulating kind of synaptic function. I suspect it's both going to be compensatory and disease directly to disease causative. I'll show you why the latter later. But you can see it's very interesting. One comment, and Kathy Morgan, can you turn off mute your computer? Okay. So that's the first step. What's also occurring in here that I'm pretty sure is important, but we're not seeing strongly yet at the moment, is I believe that this is also a lot of changes are occurring in microglia, because M6A is also linked to inflammation, but we're not yet there in terms of that level of specificity, but stay tuned. So I'll skip over this. So also, not surprisingly, when you look at genes that show changes, the MAPT gene shows changes. That's probably because the cell, Kathy, again, turn off your mute your computer. That's also, that's not surprising because MAPT is strongly overexpressed. And so the cell probably is having a response to that to like try to downregulate the gene. But also you can see other genes that are kind of involved here that are relevant to Alzheimer's disease. So it turns out RBFOX1 is strongly methylated, which is really interesting because we have another project with circular RNA that we're doing with Xiaoling Zhang and Sambhavi Puri is doing this, where our RBFOX1 regulates circular RNA. That could be a different story, I could tell. And then, of course, the glutamate receptors. So this is kind of what the volcano plot looks like, and this is what specific examples of genes in human data that are changed. And so you can see here that, like, if you look at APCA7, there's a 21-fold difference in methylation between Alzheimer and control. Mind you, this is just a set of 4 and 4. We have, just this morning, I saw Jenna putting a set of extra 10 AD and 10 control into the DART-seq protocol so that we can really get much more definitive information on this. But what you can see is that there's a strong difference in methylation and a slight difference in not much of a difference in the actual gene expression. But if you look at ABI3, another gene, you can see that again strong difference, reduction in methylation, and here correspondingly there's an increase in gene expression. So that's the kind of data we're looking for. When we don't see some difference in expression, what we think about is localization or translation. So onward to the plan for this talk. The thought was, if we have all these genes that are being whose methylation is changing with disease, then perhaps we can develop a way of detecting that in fluids. So we are progressively and iteratively developing a CSF strategy for measuring M6A. So the first idea is to take a binding protein and attach it to a solid substrate. So the first thing we've worked with is anti-M6A antibody, which we then attach to beads. But as I'll show you, we're evolving this where we're going to be moving to YTH, and I'll explain why that's important above and beyond the specificity in a moment. But anyway, so you attach the M6A to beads, you incubate it with CSF, you pull it down, and then wash it, and then do PCR on it. And the power of this assay is that it is very easy to look at different transcripts, and so modify them with disease, modify them to fit the target, because all you have to do is make different amplimers, because the pull down is always going to be the same. And then you bring in the power of PCR for doing this. So right now we're doing this kind of laborious protocol of pulling it down, washing, pulling down again, washing, pulling down again. So a number of steps involved in the PCR. But the goal is to have the capture protein just attached to a plate. And so then you do just a classic ELISA assay, and it should work. And so that's where being able to produce the YTH domain protein becomes really important. So, but even with this IP M6A PCR test, what you can see is if we use RNA from cells, looking at a S18 RNA, you can see that we can have a really nice standard curve here. And in fact, we can actually go down as far as 0.16 picograms and still have significant differences. I believe that with time, I think that the variance in the assay will go down a lot as we move away from pull down. So Sophie has done this. We obtained CSF from the BU ADC, which gave us 5 controls and 1 Alzheimer case. And we also got a set of 6 and 6, now up to 12 and 12 from Mayo Clinic. And we were, so with the 6 and 6 set and the BU stuff, we were able to do assays and again, iteratively improve the methods. At first we used cyber green, and here you can see AD versus control. And so a fair bit of overlap, but not really significant, even though there is a suggestion of a difference. I think that's where I was at last time I presented at this. Now Sophie is gone, and I should have put Sophie's name on here. But she's on other slides, so apologies, Sophie. Now we've gone and done it with a TACMAN assay. So instead of using just cyber green to randomly identify the amplimers, we use TACMAN. And with that, you can see that if you compare the control versus the AD, you really have start to get towards strong significance, even with this small set of samples, although you can see that there's a variance. And these, just for you to understand, are the negative controls, just IgG. So we've also looked at digital PCR as a way of improving it. Although I think the data are not much better than what we got for the regular qPCR. So here again, same samples, but you can see that here's the control and here's the AD. We have a nice difference, similar to what we saw with qPCR. But so we now have a set of 12 and 12 from Mayo Clinic, and we're, I think as of today, should have an extra 20 and 20 from MGH. I tried to contact NCRAD, but they felt that the data were not sufficient to give CSF. So we're kind of increasing our numbers to kind of reach the threshold that NCRAD wants to be able to donate CSF. Of the reviewers, there were 3. 2 of them said, absolutely, this is great. And one reviewer was kind of pissy and was like, well, you really need to do a lot more on this. So anyway, so what we've done now is we've taken this YTH and we've attached tags to it. First, we attached a halo tag, which is something available commercially. And the idea is to use the halo tag and use click chemistry to bind a biotin to it. We could also do click chemistry directly in the lab and bind a biotin. But Ali Rondon Ortiz, my lab, came up with a great idea of attaching an AVI tag to the YTH. And so what that does is the AVI directly biotinylates the YTH, and then you can cleave it off with a P2A domain. So with that, and we've now made this protein. So with that, we now have are able to produce in the lab abundant biotinylated YTH protein. And with that, we can start having enough protein to make ELISA assays where we just coat the bottom of a plate, or or do an alpha ELISA in solution, or use nanofibers and be able to kind of do this in a plate. And then depending on how strong it is, our goal is ultimately to be able to use like something like a Simoa platform where we're not even using PCR, but that depends on how abundant the M6A labeled constructs are in the CSF. Certainly for the RRNA, for the S18 rRNA, that's quite possible. And then another element of this that's important is just to say that not only is this kind of a biomarker for disease, but we actually think that this can modulate the disease process, because if you reduce metal 3, you reduce M6A, and you can reduce kind of the translational response to the tau. Let's see if I go, just going to skip off this. And if we take our assembloids and treat them with an M6A inhibitor, you can see a reduction of M6A. You can see a nice reduction of tau oligomerization. You can see a reduction of inflammation and increase in neuronal markers suggesting protection. And we've also now done this in vivo, where we took ALK-BH5 and transduced the gene into the hippocampus, so we could reduce M6A by overexpressing ALK-BH5, and with that, seems like we have a reduction in TOMA2 and improvement in neuronal counts. We're now in the process of repeating that, so stay tuned. We want to make sure we can repeat everything before kind of sending that out. Anyway, kind of for conclusions to this talk, we've shown you that M6A changes with disease. We're in the process of identifying the transcripts that are changed, but it appears to be strongly synaptic transcripts that are changed. I've shown you that we can detect M6A in CSF, and our preliminary studies using the 18S RNA really show a nice, like, fourfold difference between Alzheimer and control. I was really pleased at how well that worked out, and we're moving forward with this. Finally, as a little teaser, in CTAD, my company is beginning to present its Alzheimer platform. So we did screens to identify compounds that could reduce tau based on the RNA metabolism concept. And what we came up with is our lead compound, which is potent, orally available, brain penetrant, it accumulates in the brain, and even in these initial studies, we're repeating it now, greatly reduces tau in relatively late stage mice. This is treating mice at 9 months for 1 month with the compound, and the tau goes way down. So that is kind of poised to do the toxicology studies in rat and dog. We already know that it's tolerated well in rat and mice, shows very few side effects. So I think that's very promising. Anyway, and then there's the team. I want to particularly acknowledge Jenna and Sambhavi, who've done the M6A RIP-seq and the DART-seq, along with the help of Xiaoling Zhang and Junming Hu. And then the M6A diagnostic assay is entirely done by Sophie van der Spek, who's done a fabulous job on this. And of course, we can't do this without the help of ADRCs, including BU, Emory, Mayo, MGH, Puget Sound, and my other collaborators and everyone. So thanks.
Great. Been a very comprehensive and tour de force. I'm just going to open it up to questions. If someone has a question, go ahead. Or a comment.
Ben, I have a question. This M6A, so do you plan to inhibit all the methylation, or you intend to inhibit specific gene?
Therapeutically?
What?
Therapeutically, you mean?
I think it's just M6A methylation may be beneficial for some genes, but harmful for the other genes.
Yeah. Like my company Aquina was hesitant about moving ahead on M6A as a therapeutic because of the broad effects of M6A. So that's in a way why I think that the biggest I think the most immediate impact of this work is going to be for the diagnostics, where I think it has huge opportunities there, not just in Alzheimer's, in cancer. They've done a lot on M6A biomarkers, an immense amount of work, but they haven't done any serum fluid-based assays. So there's a lot of opportunity there too. They are in cancer, they are using, so Storm Therapeutics is pushing forward their metal-3 inhibitor into the clinic for lymphoma and leukemia. And for Alzheimer's, that brings up the issue, which is you might worry about it for the brain, but actually a bigger issue is the bone marrow suppression, which is a big challenge. But I think that what we do is we will test this, as far as whether it's good or bad, M6A is a blunt tool. to modulate. And so we'll just have to see. Generally, there is zero question that you need. M6A is a lot like blood pressure. You need some M6A around. If you have no blood pressure, you die. If you have no M6A, you also don't do well. But if your blood pressure is chronically elevated, that's also not good. And the same way for M6A, it's chronically elevated, it's not good. But as you point out, M6A might be chronically elevated in neurons with disease pathology, but what about in the neurons that don't have disease pathology? Probably not elevated. So, it's going to be a work in force and a work in progress in that. And the other thing is, in that context, it might be that metal 14 is a better target than metal 3, because metal 14 is kind of a milder effect on M6A, so maybe you just want to tamp it down a bit. So we'll see. But meanwhile, the drug that my company has, I showed you, is not in that pathway. It's in the ER pathway. So, I think that's more of a classic approach, and is probably going to be broadly beneficial.
I think the strategy your lab has is good to identify specific genes. I don't know why, but I was really amazed that recently, like siRNA or ASO, those kind of things are really working for central nervous system. Actually, they were not so good for cancer treatment, but very good for central nervous system. Once you identify the genes, maybe those can be a tool to target specific methylating.
You're right. That's true. I mean, you're right. Quite possible.
Very nice work. Thank you.
Hey, Ben. This is Lei. I'm a new faculty member in biomedical genetics, and mainly working on epigenetic and genetic regulation. Very interesting talk. And I have several questions. So the first question is about, maybe I missed it. So you find M6A in RNA in CSF show different signal between AD and the control. Then my question is, that RNA, is it coming from the cell-free RNA, or is it coming from certain immune cell types?
So, so the CSF that we got has been spun down. So there's kind of, when we get it, it doesn't have cells in it. However, there were cells in the CSF, and so we can exclude that the that the rRNA, we don't know at this moment whether the RNA is coming from the brain or like leaking out, or is it coming from the cells in the CSF. And there's also a question, it might be in exosomes, so that's another thing. These are all things to do. My view on the RNA is that, honestly, I was really surprised that it showed such a strong disease-dependent difference. I told Sophie to do it because I thought it would be abundant and easy to test. So it's more like a neurofilament test, like a general, I think, reflecting general disease in the brain. And so, but you're right. As we go towards, like, there's a lot of microRNA that are methylated. I bet those are going to show up. There's long non-coding RNA. All those things are things that we can really test. We just have to finish our DART-seq so we know kind of what genes to look at. Mind you, we're just looking at the brain, so then we have to iteratively go through and figure out what's actually detectable in CSF and changed. So.
Yeah, yeah, great. And I guess another question is that you also found decreasing M6A level is protected in several AD models. I was wondering, I mean, could that be the mechanism about epigenomic erosion? I mean, I think there was a paper talk about epigenomic erosion during the aging, which means that epigenetics are well maintained when during the healthy state, but during aging, then there's some misregulation. Something should be open, but then closed. Something should be closed, and then it's open. And I guess I was wondering whether you are have similar insight into that, to say maybe there was some misregulation happens. That's why decreasing M6 level can stop that kind of dysregulation.
So it turns out that aging and tauopathy, the tauopathy part of Alzheimer's, are very different things. Because if you look at an aged brain, or if you look at a brain of a mouse model expressing only APP, you actually see that disease causes disease or age cause a reduction of M6A. But in tauopathy, let me generalize this, in diseases that develop intracellular protein aggregates, this has been shown for tauopathy. This has been shown for ALS, TDP-43 diseases, and this has been shown for Huntington's disease. You actually see increased M6A. The exception to that is there's another disease type of ALS that has a mutation in C9orf72. Those develop less M6A. So again, M6A is a really blunt tool, and as Wendy was suggesting, talking about M6A generally maybe is only important when considering, using a M6A therapeutic. For any kind of diagnostic thing, we really have to think about the specific genes that are changing. And then, as Wendy suggested, though, that might help us target specific genes. But that's a very good point. As far as, as far as cytoplasmic, as far as dysregulation of M6A feeding back to regulate the epigenome, I don't know cause or effect there. So it certainly seems possible that the two are connected, but we need to do forward genetics to really answer that question, to determine which is driving which. Certainly as a general principle, and generally for most neurodegenerative diseases, there is a general principle that seems to be the case, which is that the protein aggregates that occur in all these diseases kind of sequester other proteins that cause disease. So for instance, in Alzheimer's, the intracellular aggregate is tau. In Parkinson's, it's synuclein. In ALS, it's TDP-43. Each of those have a strong tendency to aggregate, but they don't aggregate alone. As they aggregate, they pull down other crap with them. That can include many RNA and DNA binding proteins, which can then alter the epigenome. And so that's why the field has had such a strong focus on protein aggregates. Although the Alzheimer field is also these days strongly focused on inflammation also.
So Ben, if thinking about this as a potential biomarker for diagnostic purposes, so on the one hand, you've been talking the spectrum of dementias that many of them have tau connections. What about if you look at other dementias that really don't, such as vascular?
Yeah, so that's totally on the agenda. Like now that Jenna, with the help of Jenna doing the developing the DART-seq, and with the help of Sophie making the YTH protein, the YTH apobec for the DART-seq, now it becomes really easy for us to take all these AD brains and simply collect the RNA. We have a tape station in the lab, so we now just directly determine the RIN score. So we have actually, for AD, we now have a series of brains that have RINs that are kind of 7 or greater. And then go and exactly ask that question. And then as we know which genes are changed, we can then assay for them in CSF and presumably for vascular disease in blood. So yeah, totally agree.
All right. Shelling, did you have a question? You turned on your video.
She's
Yeah,
just
no, I have that very exciting work. I have one general question. So you mentioned the aging. So for aging, in aging, healthy aging brains, amyloid beta versus tau, which is more significant
for what?
for healthy aging? In a healthy aging brain?
Yeah. You mean a person who doesn't have dementia?
Yeah, don't have dementia.
My sense is that it's lipid metabolism and inflammation, isn't it? I mean, in other words, and vascular. Kathy, sorry, I see your face. I can't hear you, but I know the moment I see Kathy Morgan, I think vascular. So absolutely, it's like, I think that like tau in particular is a disease process that takes the body away from healthy aging.
From healthy aging.
And the people who have healthy aging kind of have these things, have mutations that kind of prevent, that keep their lipid metabolism good, keep their inflammation good, their vasculature good, and don't cause and essentially reduce the stress response. If I was going to talk in general, what causes tau to aggregate and what causes, first tau, what causes tau to aggregate? Stress. That's well documented, like chronic stress can induce it. Say probably also for TDP-43, that's a lysosomal stress. I'm now been given permission without giving enabling details, to begin to talk about my company's compounds to address TDP-43. And so we have compounds that can that for some of the genes completely correct cryptic splicing of TDP-43. So you can so if you keep the stress down, that's healthy aging. But as Lindsay and Kathy were hinting at, when you get old, getting enough, having good vasculature is a huge element of that.
Yeah, the reason I asked is you show M6A with tau pathology, but you don't show with M6A with amyloid beta.
Yeah, no, the data suggests that M6A correlates with two things: tau pathology, and also I've seen a lot of data. We have some data suggesting this too. I showed it to you but I don't have broad data for this. I think M6A is also important in inflammation. But A beta causes inflammation, so there's a disconnect there. But there's already published articles where people have looked at APP mice that don't have tau pathology, and their M6A is not elevated.
It's not elevated, not changed, basically.
Yeah, and we have a paper where we put out that's in BioRx. We're just finishing it. We've now fully finished all the data, we look at an APP NLGF mouse, a P301S tau mouse, and an AP NLGF cross to the P301S tau mouse, and the M6A follows the tau, much less so the A beta.
Yeah, that's interesting because Cicely is here as well. So he mentioned the epigenetics. So there is a well-established epigenetics aging marker using several hundred methylation, DNA methylation marker.
Yeah,
they
can
but for,
tell your age, your chronological age, just by looking at your DNA.
But then I'm thinking for M6A, you mentioned M6A may be similar to blood pressure, some measurements.
Yeah.
If we can use this kind of M6A IP transcriptome marker, similar to epigenetics marker, we can measure them longitudinally, some
That's it.
marker. So this is curious whether the interaction, as they said, epigenome with the IP transcriptome overall.
Yeah. And I bet actually, if we think of going to blood, I bet that the vascular dementia will, like M6A will be easier to read out vascular changes than brain changes, because that's all over blood. And M6A is known to change with heart disease. So that's something as we go forward to think about.
Okay, I want to give, is there someone else who hasn't asked a question or made a comment that would like to do so? Are there other questions or comments from anybody?
Can I add one?
Go ahead, Lei.
Yeah, I guess it's also related to Xiaoling's efforts. So I'm trying to say that for the differential M6A activity, did you also check whether there is some AD-GWAS signal there? So which means maybe AD-associated genetic variants maybe contribute to that differential signal. Thank you.
Yeah, so we had a little miscommunication. So at first I thought that the Duke people were going to do the M6A informatics, but they didn't really.
No, then we propose that in our grant. M2, yeah. After this, yeah, we have his check with the M6A QTL results. Yeah.
So it's on the works. We were literally just talking about that extensively yesterday.
Yeah.
But if you're new to BU and you want to chat with me or join in our M6A group, by all means, inclusivity. So just shoot a line to Xiaoling or me, and you can join in. Okay.
Yeah, definitely. So previously we were working on M6A and A3 ketone methylation QTL in GTEx cohort. So I guess follow Xiaoling's point, there may be some loci, their function are coupled. So maybe there is something we can do on that.
That would be great. Totally. Yeah. We meet every Monday now at 9:30, right, Xiaoling?
Yeah.
Yeah,
but unlike most informatics people, we're now meeting in person. That's the one.
Yeah, I saw. Actually, I passed by.
So that's the one. I prefer meeting in person rather than online.
Yeah, me too.
Okay, in the minute or two remaining, any other questions, comments from Ben?
Ben, I just want to have a comment that remember this my lab and also the project tool with studying vasculature, especially endothelial cell. So when we did endothelial cells between AD and control, the one protein pop out by phosphoproteomic is this protein HNRR, this RNA binding protein you mentioned.
NRPA2B1.
Yeah. So I think you talk about information and etc. I think that's something we
maybe yeah, an example, Wendy, if you give us some endothelial cells, to do the DART-seq, we need very little RNA. So Jenna, who's listening online, can really run it through, and we can do it, and so see, she's very eager. So we can do it, and maybe we'll pull up some biomarkers that will open up a whole vascular component for this. And definitely, Lei, if you have some magic to throw in on this, I'm very interested.
All right. Well, we've about reached the top of the hour. I again want to thank Ben for a very informative presentation that engendered a lot of questions, discussion, and maybe even some new collaboration. And I want to thank everybody for joining us for this ARC meeting. Please stay tuned for other messaging about the next ARC meeting and other ARC-related activities. So again, thank you, everybody, and enjoy the rest of your day.
Okay,
All
thanks.
right, thank you.
Bye.